In the late 1900s, a one-day-old boy, who we can call John, was admitted into a hospital in Cleveland. John was born with a genetic disorder that caused him to have a severely compromised immune system. This disorder is famously known as bubble boy disease because patients were often kept in these protective plastic bubbles to prevent them from having deadly infections. Now, John stayed in the hospital for about 12 months before he was able to go home and continue to receive treatment. However, during his time in the hospital at around the age of 3 months, something interesting started to happen. John was having rapid tremors in his hands, as well as strange muscular movements in his posture when he would try to reach out for objects. And John was not the only one. More and more reports were coming out of patients with the same disease who were having similar symptoms such as tremors, learning delays, hearing loss, and more. Now, this is an interesting question, especially for me as a neuroscientist. How is this disease that is not neurological in nature causing these neurological symptoms? My name is Shaima, and I'm a PhD student working at the Douglas Mental Health University Institute. I'm studying one particular form of bubble boy disease, which is known as ADA-SCID for short, or as we scientists like to call it because we love to complicate things, adenosine deaminase deficient severe combined immunodeficiency. So the different types of SCID get their names depending on the genetic defect that caused them. And the particular type that I'm studying, which is ADA-SCID, is called that way because the affected gene is called ADA. And the question that my research focuses on is, how is the genetic defect of ADA-SCID causing these neurological symptoms in patients such as John? And basically, how is it affecting the nervous system? So now that we know what the question is, why isn't it easily studied, and how am I trying to study it? Well, it's not easily studied for two reasons. The first reason is that by the time that these neurological symptoms appear, the damage to the brain is already done, so to speak. And it's difficult for us to know exactly how it started. The other reason that makes it difficult to study this disease and other diseases that affect the brain is that we can't safely obtain a sample of patients' brain tissue because it's an invasive procedure, and I don't think anyone would want that done to them, and it can further damage the brain. So it's possible to do it in the case that a patient is already undergoing some kind of surgical procedure, but that's not always the case. So what do scientists do? Well, they either use animal models or post-mortem tissue, or they perform imaging scans on live patients to try and understand how a disease is affecting the brain. And while this has provided us with some information in terms of ADA-SCID, it doesn't allow us to understand how the disease is affecting the brain starting early in development, which is really important, and also how it affects the way that cells function. And this is where my research comes in to try and answer these questions. When I first became a neuroscientist, something that was really exciting for me was using stem cells in order to study diseases. So this is exactly what I'm doing. I'm using stem cells in order to study how the genetic defect of ADA-SCID affects the brains of patients and causes these symptoms. And this is a really exciting thing because even 50 years after the genetic cause of SCID has been discovered, we still don't have the answers as to how it affects the nervous system. So I hope that my research could try and answer that question. Now, what are stem cells? Well, you can think of stem cells as the baby cells that can then grow up to become any type of cell in the human body, such as your skin cells, your heart cells, brain cells, and much, much more. Literally every single cell that makes up your body. And these types of cells, like skin cells, for example, can be considered or thought of as mature cells. And in the human body, the cells would naturally follow the development from their baby state to their mature or grown-up state, just like us, basically. So how do these stem cells know what type of cell to become, given that they have so many options? They receive chemical messages which tell them what type of cell to become. And these messages depend on where the stem cell is located and also what surrounds it. And once the stem cell receives that message, it can basically switch off its genetic recipe for being a baby stem cell, and it can switch on its genetic recipe for being a specific mature type of cell. Now, the great thing about scientific research is that we have access to biobanks, which include biological samples from patients who have different types of diseases, even those who are unfortunately no longer with us, which is a great tool for us to use. So I obtain skin cells from ADA SCID patients. And what I can do is I can use genetic engineering to essentially rewind them back to their baby state as stem cells. And this is really, really useful because now we can have access to stem cells from patients without necessarily getting them from an embryo. Now that I have these baby stem cells, I can then make any type of cell in the lab using, basically by mimicking the same chemical messages that stem cells receive in the human body that tell them to become specific types of mature cells. And one thing to keep in mind is that every single cell that we will then make will carry the same genetic defect that these stem cells from patients have. So I can specifically ask the question, how is this genetic defect of ADA SCID affecting the way that brain cells develop and the way that they function? So to answer the question of how it affects the way they develop, what I make is an intermediate type of cell, which is called a neural precursor cell. And I can try and discover what kind of changes the disease is causing in these cells. And once I can discover that, I can then make specific types of mature brain cells, like neurons, glia, and other types of cells. And I can discover how these changes are affecting the way that they function. And one thing that you may have heard of before is that in scientific research, we need to have some sort of comparison between two groups for us to be able to make a meaningful conclusion. And in my specific case, the two groups are ADA SCID patients and healthy individuals. And I'm lucky enough that through the same biobank, I can actually get skin samples from the healthy parents of these patients. And I can then perform all the same kinds of experiments on these cells to be able to make meaningful conclusions. So to put things into perspective, we have this immune disease which is causing these neurological symptoms that have been a mystery for decades. And now, using the power of stem cells, we actually have the tools that we can use in the lab to try and discover what mechanisms are actually causing this. So as more and more discoveries are being made and advances are being made for treatments for the immune side of ADA SCID, I hope that my research can also help us understand the neurological side of the disease so that we can also find treatments for those symptoms. And in doing that, patients like John would not only be able to survive outside of a plastic bubble, but like all of us, they can get a chance to thrive in all aspects of their lives. Thank you.